Namaste friends, welcome back to the biology session. Let us continue with the chapter digestion and absorption. This is Manjunath from Sankalp CET course. We were discussing about salivary gland, right? In that salivary gland, we discussed the largest one is parotid. Largest one is parotid, which is the largest, and that is situated in the cheek, right? Cheek, I can say that below and in front of the ear, below and in front of the ears, both sides. All these are paired. So let us take that the question related to that, the location, and same like that sublingual below the tongue, right? And then submaxillary, that is in the lower jaw, at an angle of lower jaw. Match the following column 1 and 2. So parotid, as we know that is present seen in the cheek, that is below and in front of the ear. We can also take that below and in front of the ear, right? So that is A3. So first what you should do, you have to find out that is there, in which option we can see A3. See here only in the fourth option we can see that A, which has the correct match with the 3. So now we can select that, but let us check once again. Submaxillary, submandibular. Maxillary salivary gland is also called as mandibular. Upper, the bone, right, which is maxilla, and lower, which can be movable, that is mandible. So lower, the angle, that is B2. And same like that sublingual, that is below the tongue. So A3, B2, C1. So what is the correct answer? That is fourth option is the correct answer for 12th question, right? So parotid, that is in cheek internally, or you can say below and in front of the ear, and submaxillary, which is also called submandibular, at an angle of lower jaw, sublingual below the tongue. We know that lingual, sometimes we'll say linguistic, right? I can say that the linguistic, or I can say bilingual, so that is related to tongue, right? So answer is fourth one. Next, saliva, which is produced, right? We already discussed that is pH, which will be 6.8. So pH, which will be 6.8. So answer for this, the pH of saliva, that is the enzyme which will be there, salivary amylase, right, which helps in the digestion of starch, right? And then we can also see lysozyme, which kills the bacteria, right? Antibacterial, act against, or which kills the bacteria, right? Same like that we can also say mucin will be present, that which helps in the mainly with mucus, which helps in binding food particle. So these are the things which are present in saliva. So saliva produced by salivary gland. So next is that next question. Before going to take next question, right, as we all know that alimentary canal that opens, that is the first the anterior part, mouth opens into the buccal cavity, and the buccal cavity which has oral cavity, and in that oral cavity we can see palate and tongue, correct, right? So palate and tongue, upper palate and lower tongue. So now this buccal cavity which opens into the oropharynx or pharynx, right? So what exactly the pharynx? So if you consider this structure, right, so pharynx which opens like this, right? Here, this is very important. Two terms we're going to present the, right? Here there will be an opening. So this region, which is called as nasopharynx, and that is from the, what is it, nasal passage, and from the oral cavity, and this is the, what is it, oral cavity. So oral cavity which opens into the oropharynx. What is it? Oropharynx. So now this oropharynx opens into the pharynx, right? So this is very important. So pharynx, right, which receives the opening from the lower side. If you're going to say that this is the, what is that, which receives the food pipe. So food pipe, which is also called as, we know that, esophagus, or which opens into the, from, what is it, the upper end, we're going to say, which opens into the, what is it, esophagus through an aperture. This is an aperture called as gullet. Gullet is an aperture, an opening, an aperture which opens into the food pipe, Clear there? Food pipe is also called as esophagus. And here, the opening which is from the pharynx that opens into the trachea, which is lined by C-shaped hyaline cartilage, which prevents from collapsing. So that is the opening, that is the opening of the pharynx into the trachea, or we can say windpipe. Trachea, which is also called as windpipe. So now that opening, right, which is called as, what is it, glottis. So one is gullet, you have to keep it in mind. Other one is glottis. And this glottis, which is, what is that, covered by a plaque-like, what is it, the structure, the plaque-like structure, right? So that is the epiglottis. What is that? Epiglottis, which prevents the entry of food into the glottis, or we can say through glottis into the trachea, So this is, you have to keep it in mind in case of pharynx. And pharynx, that which in turn opens into the esophagus. As we are discussing about digestive system, let us take esophagus. So esophagus, where no digestion takes place, because no, what is it, enzyme producing glands. No enzymes will be produced in esophagus. So no enzyme producing glands. That is what we can say, no enzymes, because of the absence of digestive glands in the esophagus. Digestive glands in the absence of digestive glands in the esophagus. Now let us take next continuation, right? So this is where exactly the goblet cells will be there, right, to make food easy movement. Esophagus, right, which will be with no, what is it, the digestive glands, but there will be with, what is that, goblet cells. So these goblet cells, as we all know, that these will going to produce mucus, which helps in lubrication of food. So now this esophagus in turn opens into, what is it, further opens into the stomach. So let us take the structure of stomach here. Stomach is J-shaped pouch-like structure. Just one rough diagram we can have. This is the esophagus that opens into the J-shaped stomach. Right? And this is the duodenal part. So this, what is it, the J-shaped stomach, where esophagus, this is the esophagus, where it opens actually into the, what is that, mainly to the cardiac region. So this region of the stomach, which is called as cardiac region. So very important, that opens to the cardiac part or cardiac region, and this region which is called as fundic region or fundus or body, right? So, and this region which is called as pyloric region. Pyloric region. So, here the very important thing you have to keep it in mind that that is the fundus, cardiac, pyloric. So, as it opens, the opening will be there in the the fundus region, and here it is guarded by a sphincter, guarded by a muscular sphincter, right, which is called as mainly cardiac sphincter, because as it is opening into the cardiac region, hence it is called as cardiac sphincter, which is also called as esophageal cardiac sphincter. Cardiac sphincter, which is also called as esophageal, which is also called as esophageal cardiac sphincter. Esophageal cardiac sphincter. So you should keep it in mind this, right, cardiac sphincter. And here the opening of the pyloric region into the duodenum is guarded by a sphincter called as, right, that is a muscular wall which prevents, here it prevents the backflow, here it prevents the backflow, what is that, from the cardiac region, or you can say from the stomach into the esophagus, right. And here it prevents, it helps in the pyloric sphincter mainly, the function as regulation of movement, regulation of food movement. Here it prevents the backflow, it prevents the backflow, right, that is the function of the cardiac sphincter. And here it regulates the movement of food into the duodenum, movement of food, right. So that is what you have to keep it in mind. And in stomach there are the specialized cells. Those cells will consider as generally different types of cells which present in the glands. They are going to secrete the chemicals, like main the cells of the stomach, Auxintic cells, right, we can say Auxintic cells or parietal cells, parietal, which are also called as Auxintic cells, which produces HCl. This is very important. Auxintic cells or parietal cells produces, and then peptic cells, or which are also called as zymogenic cells. Peptic cells or zymogenic cells which produces pepsinogen, which are in the inactive form. Pepsinogen, or we can say zymogen, both which will be helps in the digestion of protein, but they are in inactive form. HCl is needed for the activation of those the pepsin, mainly for the conversion of pepsinogen into pepsin. So next, the one more, that is there are the the hormone producing glands, hormone producing endocrine glands, we can say G cells, which produces gastrin, which produces a gastrin, that is hormone produced in the stomach, right. Now let us take what is the role of the parietal cell once again, that is which produces HCl, right, and also peptic cells, zymogenic cells, which produces pepsinogen and zymogen, right, and G cells, gastrin. And here parietal cells which will be going to produce HCl, along with that it also produces Kessel's intrinsic factor, so which helps in mainly the digestion of, this is very important, Kessel's intrinsic factor and vitamin the B12 together, right, mainly in the absorption of vitamin B12. So both together which helps in the HCl helps in absorption of iron, conversion of Fe3 plus into Fe2 plus, and this vitamin, and this the Fe2 plus, that is ferrous ion and vitamin B12 together helps in the synthesis of RBC. Hence Kessel's intrinsic factor and HCl play a role in the production of RBC. If no Kessel's intrinsic factor or HCl, that leads to what is it? Pernicious anemia. What is it? Pernicious anemia means no, what is it? RBC's production will be there. No, what is it? Red blood cells production. So this is very important. Kessel's intrinsic factor, right? Mainly produced by Auxintic cells, right? Parietal cells, Auxintic cells, peptic cells, which are also called as zymogenic cells, right? So next we'll take what is it? The liver. So now before going to what is it? The different parts of the liver, just we should keep it in mind that is the two-lobed, that is the externally we can say that is two-lobed structure, and where in the what is it, the mainly the liver in the posterior region there the gallbladder which will be attached to the posterior region, right? And this gallbladder opens into the cystic duct, and here is the right hepatic duct and here is the left hepatic duct, and those join to form what is it? Common hepatic duct. So this is what is that? We can say we can say this one which is right hepatic duct, and this one is left hepatic duct, both join together to form what is that? Common hepatic duct, and this is a cystic duct of gallbladder is called as cystic duct, joins to form what is that? This is the very important thing, common bile duct. Why the name is called as common bile duct? It will going to what is it? They carry bile juice which is produced from the liver, and also that bile juice stored in the gallbladder. Gallbladder is a structure where it is present seen in the posterior region of the liver, which helps to store bile juice. Which what is it? Stores bile juice. So these are the a very important thing in the liver. So liver and further what is it? The structure we can discuss, right? So let us take some question. Here liver, the main function of the liver, right? So just I will mention here the main function of the liver is the production of bile juice. So bile juice which is very important for emulsification of fat. Right? So this generally liver is covered by, if a mammalian liver, if you're going to say that, outer visceral peritoneum will be there, and inner what is that? Is covered by outer visceral peritoneum. And one the characteristic feature of the mammalian what is it? The liver is it is covered by Glison's capsule. Next to that inner Glison's capsule. So just to represent that is we can say Glison's capsule is a characteristic feature of mammalian liver. So we can expect this question, right? Next, let us take some question what we have discussed till now. Identify the false statement. Esophagus does not secrete any enzyme. Correct, right? As there are no glands, no enzymes. No glands, no enzymes. That is a correct statement. We want false statement. This is a correct statement. Gallbladder present in human being. Just now we discussed gallbladder which helps in what is that? Bile juice, which helps in storage of bile juice. Stores the bile juice. Human teeth are thecodon, means embedded in bony socket. Embedded in bony socket. A very strong, right? They are different types. heterodont and 2 sets diphyodont. Heterodont and 2 sets in the lifetime, hence they are called as diphyodont. Diphyodont. There are 2 pairs of salivary gland. Is this correct? No. There are 3 pairs of salivary. The false statement is D. That is 3 pairs are parotid, sublingual, which are also called as sublingual, and the third one is submaxillary or submandibular, right? It is parotid, sublingual below the tongue, and submaxillary. Submaxillary, or we can say submandibular. Submandibular are the 3 pairs. There are totally 3 pairs. Next, a gland which is not associated with the alimentary canal. So as we have mentioned, it includes a pancreas and liver also included, salivary gland, pancreas which produces a pancreas juice and certain hormones, hence it acts as heterocrine gland. Hence it acts as heterocrine gland or mixocrine gland, right? Both exo and it acts as both exocrine. It is both exocrine and endocrine in function. Exocrine, acinar cells, acini, many. Endocrine, islets of Langerhans, islets of Langerhans, right? So acini, which are the cells which produces enzymes, mainly 3 inactive enzymes, let us take within a while, and islets of Langerhans, which will going to produce hormones, So this is very, very important. Next, and in liver, if you're going to say that, we know that there are the hepatic lobules, which are the structural and functional units in the liver. Hepatic lobules. In that lobules, the structural and functional units are hepatocytes. So hepatocytes means liver cells which mainly produces bile juice. So next. So what is the answer for this? A gland which is not associated with alimentary canal? Adrenal gland. Right? We know that salivary gland produces saliva. So next. Structures that open into a pharynx. They are asked structures. So if structure that open, that is the buccal cavity, oral cavity, in which, what is that? We can take this diagram once again. See, nasopharynx and oropharynx together, which will going to open into the pharynx, right? But in the option what they have given, let us see, right? The structures, see, they have given trachea and esophagus and epiglottis, right? So if you consider this is a pharynx and that which will going to receive, right, the trachea, the structures that open into pharynx. One is the trachea which opens, other one is esophagus. Through, what is it, the plinth-like structure that is different, that is mainly we can say that is trachea, that is a windpipe, and food pipe. Esophagus from the lower side that will going to open into the pharynx region. So answer is both 1 and 2. Right? So epiglottis which opens into the, what is that? Glottis, which covers the glottis. It is not a structure, it's a flap, which is an aperture. It is a flap-like structure. Next, epiglottis, here only it is there, cartilaginous flap-like structure, which prevents the entry of food into. So epiglottis is the cartilaginous flap. So if you consider this as a glottis and this is the windpipe, and ear, which is a flap like structure that covers. So that is mainly going to, the gullet. We know that opens, an aperture opens into the pharynx, and that is the gullet which opens from the pharynx, we can say from the pharynx into the esophagus. Esophagus, we can say like that, pharynx which opens through gullet into the esophagus, right? So that is, we can eliminate these two, gullet and esophagus. And the epiglottis, that mainly prevents the entry of food into the glottis, right? So this is a glottis, the opening, an aperture that opens into the trachea. Serosa is muscular layer, outermost muscular layer, serosa, and next to that is muscularis, right? Serosa, muscularis, submucosa, mucosa. So if you're going to, we'll take that in the later part. So that is what you have to keep in mind. What is the answer for that? Which mainly, prevents the entry of food into the glottis, and then glottis into the trachea. Next, as we all know that stomach, which mainly produces the gastric juice, so that the gastric juice which consisting of the mucus, HCl, Kessel's intrinsic factor, pepsinogen, and along with that one more, that is, we can mention renin. So this which helps in degrading milk protein called casein, which helps in the digestion of casein, mainly in the infants. That too in the herbivores, generally we can say, right, where calf, right, you can say young one of cow, right, calf, and we can say some herbivores, right, in which we can see more the renin production will be there. More renin production will be there. So hydrochloric acid in the stomach is secreted. Generally we know that this will be secreted in the stomach with cells. So as the enzymes play a major role, enzymes which play a major role, hence the enzyme producing cells are also called as chief cells. Enzyme producing cells are also called as chief cells. What are those? Peptic cells. So we can, peptic cells. Peptic cells. We can eliminate chief cells here. They involved in the the production of enzymes, not HCl, and parietal cells, which are also called as auxintic cells. They helps in the production of HCl. So these are the cells which are involved. The answer is second one. Zymogen, peptic cells, which are also called as zymogenic cells. They produces zymogen enzyme. So this is also we can eliminate. None of this. So correct answer is second option. And 19th. So here a very important thing, the cardiac sphincter protects the backflow, right? Already you mentioned, that is the cardiac sphincter which opens into the esophagus, opens through the cardiac sphincter into the cardiac region of the stomach. That is where we can say it prevents the backflow. Whereas pyloric sphincter, it regulates the movement. Further in this region, it regulates the movement of food, in from the pyloric region into the duodenum, that is small intestine. So here it is guarded by pyloric sphincter that regulates the movement. So cardiac sphincter, that mainly prevents or protects the backflow of food from the, because it is mixed with HCl, right? If it is going to come back to the esophagus, if there the backflow that occurs, that will going to erode the esophagus, erode the lining of the esophagus, but there will be mucus. That is why we are going to get the heart burning sensation, right, and cough. So that is what it prevents the backflow of food into the esophagus from stomach. We can say that is from stomach. Acidic food prevents the backflow of acidic food. So answer for that is, what is that? Fourth one. Next, milk protein casein is coagulated by. So here we can say the casein, which is a milk protein, which will be converted into para-casein in the presence of renin, and from that will be converted into calcium para-caseinate, so then we can say that curdling of milk, so that where we can say digestion can be seen. So that is, we can say curdling of milk, right, that is coagulation, breakdown of the protein, right. Next, so renin, the milk protein casein coagulated by renin, right. So proteins which will be, what is it, the digested with the help of pepsin and trypsin, but the milk protein mainly, that is renin. Next, so here one more, renin, which is also called as chymosin, So let us move on to the next question. Match the column 1 and 2, right. So peptic cells, we know that peptic cells, which are also called as, right, should repeat, that is chief cells, because they produce enzymes. Enzymes play a major role in digestion, right. That is what pepsinogen and what is it, reninogen, we can say prorenin. So these pepsinogen, if you're going to use pepsinogen, these are the inactive, and prorenin is also inactive, what is that, enzyme, that will be activated with the help of HCl. So now chief cells, which mainly produces proenzymes. What are those proenzymes? They are the pepsinogen and prorenin. When they will be converted into, what is it, the active form, when they acted upon by, what is it, HCl, they will convert it into active form in the presence of HCl. Now next part, the auxintic cells, which are also called as parietal cells, we can remember like this, HCl. And then goblet cells, we know that generally in the, what is it, the esophagus, right, mainly it helps in the production of mucus. So alkaline fluid, which is actually the secretion of small intestine, when it will be the secreted by, I can say, small intestine or pancreatic juice, we can say alkaline fluid, right. That is a secretion of those, what is that, pancreas and small intestine. So what is the answer? A3. So we can see that here there is no, what is it, the A3. Now here it is the answer. Next, let us check B4, correct, goblet cell 1. So answer is D. Right. ಇಲ್ಲಿ ನಮಗೆ ಈ ಥರದ ಆಪ್ಷನ್ ಬಂದಾಗ ಫಸ್ಟ್ A3 ಇರೋದು ನೋಡಬೇಕು. Sometime there will be with 2 options, so that we should not worry about other 2 options, we should eliminate that, right. So A3, if it is there, you have to check with these 2, right, then you can get answer and you can save time. So liver of man is, already I mentioned that liver of man, which is mainly with the 2 lobes, right lobe and left lobe, in that generally we'll say right lobe and left lobe. This is the right lobe and this one is the left lobe. So in this you have to keep it in mind, generally that is the just depression, where we can also see that the other 2 regions in the right lobe. In the right lobe we can see that is, this is called as caudate caudate, and this is called as quadrate lobe. Quadrate. So in this one, let us take one extra information, where to the quadrate region, in the quadrate region we can see that is attachment of the to the quadrate region, posteriorly to the quadrate region we can see the attachment of, what is that, gallbladder, which stores bile juice. It won't synthesize. Liver going to synthesize through cystic duct, that will going to get filled with, and the same it will going to empty into the common bile duct. Next, we'll take up the duct system later. So that is 22 bilobed. Liver of man is bilobed, and mammalian, what is it, the liver, the characteristic feature is presence of Glissen's capsule. Next, which of the following is a structural and functional unit of liver? So if you're going to see the structure, generally these each, what is it, the will be with hexagonal shape, that is the hepatic lobules, and there will be a central vein. This is the central, what is it, the vein, and surrounding that there will be the arrangement of hepatocytes in the form of cards. Hepatocytes which will be arranged in the form of cards. So this in between there is a space. This space which is called as, what is it, sinusoid. That space which is called as sinusoid, mainly, what is it, the structures which are present in that sinusoid, right? Some larger, what is it, the cells which will be there, which play a major role. The space is called as sinusoid, and those cells which are present seen, right, which are phagocytic in nature, they are called as Kupper cells, or you can say Kupper cells. So, and along with that there will be fat storage cells. They are called as, what is that, adipose cells. They are called as adipose cells. This thing you have to keep it in mind in the liver. Adipose, what is that, the cells which store, what is that, the fat, or you can say that fat cells, right? So that is what you have to keep it in mind. Next, so in each, what is the corner, hepatic portal vein opens. We'll take up that in the next, hepatic portal vein. So here you have to keep it in mind, hepatic cells means hepatocytes. They are present within the lobules, but such lobules are the, what is that, structural and functional unit of the kidney. If they won't give hepatic lobules, you can select hepatic cells, right? Hepatic cord means what? Hepatocytes arranged in the form of cords, right? Hepatic lobe, that is a complete one single lobe, right? So that is, we can consider many lobules with generally the that is, we can say hepatic lobules, right? That is hexagonal lobule, or called as hepatic lobule. Hepatic lobe means liver lobe, that is 2, right? Next, answer for the 23rd question, which of the following structural and functional unit of liver? That is hepatic lobules. Clear there? Hepatic cells illa, right? So hepatic lobes, no. Hepatic cords, that is also wrong. Next, Kupper cells. I've already mentioned that they are only, which are phagocytic in nature. What is that? Phagocytic in nature means which engulf, what is that, the foreign particle, microbes. So they are present in the sinusoids, the space present between cards, space between, what is that, the cards, hepatocards, which is called as sinusoid. In that sinusoid, that is in the liver, we can see Kupper cells, right? That is what you have to keep it in mind. And next, the function of HCl of gas structures. which mainly helps the conversion of pepsinogen. Already we discussed. So pepsinogen into pepsin, that where this pepsin can act on proteins, right? So that it provides the acidic medium where what is that pepsin can act on proteins. So that is the major role, and along with that additional, the secondary role is it kills microorganisms. But the primary role, the function of HCl in the gastric juice, that is the providing acidic medium for what is that pepsin to act on protein, and it also helps in the conversion of pepsinogen into pepsin. Right. Next question. Pancreatic juice and hormones of pancreas are produced by. So here we already mentioned that the pancreatic juice, that is with enzyme produced by what is that acinar cells, that is exocrine cells. So exocrine part which produces what is that pancreatic juice, whereas pancreatic juice will be with pancreatic enzymes. So enzymes are produced by exocrine part, that is they are acinar cells, and endocrine part islets of Langerhans. So endocrine, we all know that alpha cells which produces glucagon hormone, and then beta insulin, right, and then delta somatostatin. What is that? Statin. So this which will going to affect both what is that alpha and beta what is that the cells, prevents the production of glucagon and insulin. So pancreatic juice is produced by acinar cell, and hormones produced by endocrine cells, right, that is alpha cells, beta cells, gamma cells. Same cells? No. Same cells at different time? No. Statement is wrong. No, statement is correct, right? Next, what is that? Different cells. So pancreatic juice and hormones produced by different cells, right? Yes, that is pancreas with the exocrine cells, pancreatic juice with exocrine cells, and hormones with endocrine cells, right? Hence the pancreas is what is that heterocrine gland. Next question. Jaundice is a disease. See, liver which mainly helps in when the degradation of RBC takes place, degradation of hemoglobin, better to say, right? During that time, bile pigments will be produced. So bile pigments should be eliminated from the body. Those bile pigments are two types, that is, one is bilirubin and biliverdin. So these are the pigments which will produce during the degradation of hemoglobin, and if they accumulate, if they won't eliminate through bile juice, they should be excreted out through bile juice and then excreted out as a digestive waste. Keep it in mind, this is very important. Digestive waste, right? Digestive waste. Whereas a little part of this will be converted into urochrome. That is the pigment which will be eliminated through urine. Right? So what is the answer? If it accumulate, this because of the because of the liver, was that the cirrhosis, not kidney, that is because of the damage, or where liver, what is that, they won't perform its function, right? Damaged condition which won't generally eliminate because it won't produce bile juice, and bile juice is required for the elimination of these two pigments, right? So through bile juice it will be excreted into the what is that, mainly into the duodenum, and then through the digestive the tract it will going to eliminate all. So that is what jaundice. If liver is not perform its function, then that leads to, right? So damaged condition of liver is called as jaundice. Right. Next question. The secretion that mix with food in the small intestine are. So as we know that duodenum, which receives through the hepatopancreatic duct. I will write in a short form, hepatopancreatic duct, that which receives a common bile duct. We already discussed that. Here the pancreas, which opens with the smaller and the larger ducts, right? There are the ducts called as ducts of Wirsing and ducts of Santorni in the pancreas. So these will going to receive, right? That opens. One is, this is the duct of pancreas. So duct of pancreas and the common bile duct join to form hepatopancreatic duct. So that opens into the duodenum. Hepatopancreatic duct opens into the duodenum, and that opening is guarded by a sphincter called as sphincter of Oddi. Right? Sphincter of Oddi. Right. So that sphincter, which will going to regulate the opening and closing with the the hepatopancreatic ampulla. There is a small swelling will be there in this region, that is hepatopancreatic ampulla, right? Hepatopancreatic ampulla, right? So this which will be there in the sphincter of Oddi. Now it receives, one is bile juice. Here from the the common bile duct, it receives bile juice, and from the pancreas, right, it will going to receive pancreatic juice, right? That is pancreatic juice. And in the intestine we can see, right, the intestinal juice. In the intestinal juice. So what are those? Mainly, that is, as the food which enters, but mainly from the stomach, which enters only little quantity, and that will be converted into alkaline because of the pouring of this bile, pancreatic juice and intestinal juice. So secretion that mix with the food, that food will be with HCl content, but later when you're going to mix, that is in the small intestine mainly, which receives hepatopancreatic duct, where it has a bile juice, pancreatic juice and intestinal juice. So answer for that is bile juice. Clear is there? Next, digestion of fat is mainly facilitated by, so already we have mentioned that the bile juice which will be produced, bile juice will be with different types of bile salts, that is sodium bicarbonate, sodium glycocolate, and sodium taurocolate. So these are the salts, that is the bile salts, which will be there in the bile juice. These which will going to helps in the emulsification of fat, that is conversion of fat, what is that, into fat globules. So bile juice helps in the digestion of fat, mainly in the process of emulsification, and later that the emulsified fat, which will be converted into fatty acid and glycerol with the help of lipase. This is very important. Lipase enzyme generally produced by the pancreas, and also will be little amount will be there in the intestine. So this lipase, right, will be activated by bile juice, right? Keep it in mind, it activates the bile juice, activates the lipase. If no bile juice, no what is it, the fat digestion takes place. So, and one more thing, it is not only helps in the emulsification, it also helps in the activation of lipase. It activates the lipase. So strongest lipase is pancreatic lipase, or which is also called as steapase. Steapase, pancreatic lipase or steapase. The next question is the very important in this: the cells of the pancreas is not digested by their own enzymes. Not digested by their own enzymes, because as we all know that the pancreatic, what is it, the juice mainly, what is it, the consists of 3 proenzymes. In the pancreatic juice we can see 3 proenzymes. I have already mentioned what is the meaning of proenzymes. That is, they are inactive enzymes. That is, they are inactive enzymes. Proenzymes means which are inactive form. That inactive enzymes are trypsinogen and chymotrypsinogen and one more, procarboxypeptidase. This is very, very important, right? These are the 3 inactive enzymes. Almost all, what is it, the inactive enzymes, almost all are inactive enzymes. Among these, the 3 major enzymes which are in the inactive state are the produced by the pancreatic acinar cells, that is exocrine gland, trypsinogen, chymotrypsinogen, procarboxypeptidase. Now these which will be converted into active form in the presence of enterokinase produced by the intestinal glands. Enterokinase, note, enterokinase is non-digestive enzyme which won't helps in the digestion of food. It is non-digestive enzyme, even though it is produced by the, what is it, the digestive glands, but won't involved in digestion directly, right? It won't involve, right? Not involved in digestion, right? No involvement in the digestion. But what is the major role of this enterokinase, which is also called as enteropeptidase, produced by intestinal glands. So these are produced by intestinal glands. So these glands which produces enterokinase in the duodenum, that will going to pour, when these will be received from the, what is it, the hepatopancreatic duct, there they will going to get activate. There they will going to get, what is that, activate. So this is very important. Enterokinase is an enzyme which activates a trypsinogen mainly. Later what happens, this trypsinogen which will be converted, right, mainly enterokinase acts on trypsinogen which will be converted into trypsin. Now the trypsin, which in turn activates the chymotrypsinogen and converts the chymotrypsinogen into chymotrypsin. And same like that converts procarboxypeptidase into carboxypeptidase. That is why we'll say, in turn, when it activates the, what is that, the trypsinogen, when the trypsinogen is activated and converted into trypsin with the help of enterokinase, and then that enterokinase won't play role in the conversion of chymotrypsinogen and procarboxypeptidase directly. So now this trypsin will going to act and convert chymotrypsinogen into chymotrypsin and procarboxypeptidase. So with this we can say cells which won't, cells of the pancreas won't digest their own enzyme because these cells which are secreted, these cells which secrete enzymes, those enzymes are secreted in the form of inactive, right? So that is why the cells of pancreas won't get digested by their own enzyme, right? So when they get activated, they're going to reach in the pancreas. There won't be a lot of mucus. They're going to reach to the duodenal loop, right? So when they're going to reach to the duodenal loop, right? So here we can say this. I'm going to show here. When they're going to reach the duodenal loop, large amount of the mucus lining will be there. As soon as this enters, there will be a substrate. On that substrate, they're going to act and convert the substrate into products. Converts the substrate into products. So next question. So answer for that is A. Enzyme secreted in inactive form. Right. Cells are lined by mucous membrane. No, that is mainly the cells are lined by mucous membrane only, but largely they won't secreted in the inactive form. If they're going to secrete in active form, they may erode, may affect the lining. Next, enzymes are released only when needed. No, there will be enzyme when the food which will going to keep on moving, enzymes will be produced, but they are in the inactive state, right? And the next question you can take, that is refer the given flow chart and select the correct option what Y represents and Z represents. So what is that? Milk casein in the presence of renin, that is also called as chymosin. Renin is also called as chymosin. Here keep it in mind that one difference I will give. Renin is the hormone produced by the kidney. Hormone mainly produced by the JG cells, juxtaglomerular cells. Juxtaglomerular cells of the kidney produces renin, whereas renin is the NNIN, renin andre ado enzyme. This is very important. So NIN is a hormone and then renin, right? Illondu simple technique nodva. Generally ನಾವು ಬರಿಬೇಕಾದ್ರೆ renin, see ಹೀಗೆ ಕೂಡ್ಸ್ ಬರ್ತೀವಿ, right? Renin. So just you can turn like this or like this, that is E, enzyme. So some people will write renin like H, only one single N, that is the hormone, right? So renin will write like this. Sometimes we're going to write, people used to write like this, right? So that is what E, enzyme, right? So that is the one technique you can easily remember, right? In this case, what you should do, whenever the two terms of which are similar, just you have to they remember only one clearly. Then opposite to that, you can aramse mention that. Next, so renin, which converts a milk casein into para-casein. We already discussed this. And then in the presence of calcium ion, which will be converted into para-caseinate, that is what? Curdling of milk. So what is Y represents? Renin. And what is it? The calcium, that is Z represents the calcium ion. So this is what you have to keep in mind. So answer for that is A, right? So with this we can conclude this part, and let us take in the next class we can discuss about the other concept in the part 3, right? So in the part 3 we can discuss about the digestion of carbohydrates and lipids, proteins and other related questions. Thank you.